Welcome back to Inside Biology. I'm so excited to have you here again as we dive deep into the fascinating world of life sciences. Whether you're a student, a curious mind, or a fellow biology enthusiast, there's something here for everyone. Today, we're unraveling the building blocks of life. Proteins. Let's break down how they are made and why they're so incredible. Proteins are tiny building blocks that do big jobs in your body. They're made of smaller parts called amino acids, like beads on a string. There are 20 types of amino acids. Amino acids are molecules that combine to form proteins. Amino acids are the building blocks of proteins. When proteins are digested or broken down, amino acids are the result. Let's illustrate the structure of amino acids. Amino acids are like tiny building blocks with 4 key parts. 1. The amino group. The NH2 group. 2. The acidic group, the COOH3, the hydrogen, the H, and 4th, the variable group R, which gives specific properties to proteins. All these parts connect to the same carbon atom, called the alpha carbon, which is why they're named alpha amino acids. Next, we will explain the amphoteric nature of amino acids enables them to function as buffers. Amino acids are amphoteric because they possess both acidic and basic functional groups, allowing them to react with both acids and bases. Specifically, they have a carboxylic acid group that can donate a proton and act as an acid, and an amino group, NH2, that can accept a proton and act as a base. Here's the magic. In neutral pH, like your blood, it becomes a zwitterion, with a positive NH3 head and negative COO tail. In acidic medium it grabs extra protons like a sponge, and in basic medium it donates protons like a charity. This dual ability makes amino acids effective buffers, helping to maintain a stable pH in biological systems. For example, proteins, which are made up of amino acids, play a crucial role in buffering blood pH. Next, we will illustrate the condensation, dehydration, synthesis, and hydrolysis, breakdown, of peptide linkages. When amino acids link together to form proteins, they undergo condensation reactions, also called dehydration synthesis. In this process, the carboxyl group of one amino acid reacts with the amino group of another, releasing a water molecule as a byproduct. A strong peptide bond is formed, creating a dipeptide. Repeated over and over, this reaction builds polypeptide chains, the backbone of proteins. This assembly requires energy, ATP, and is often facilitated by enzymes, like those in ribosomes. Proteins are broken down into their constituent amino acids through hydrolysis, a chemical reaction that involves the addition of water, HO, to break peptide bonds. This process is essential for digestion, recycling cellular proteins, and regulating biological functions.
Coming up next is the difference between essential and non-essential amino acids, acidic and basic amino acids, polar and nonpolar amino acids. Starting with essential and non-essential amino acids. Your body needs 20 amino acids to build proteins, but here's the catch. While it can make some called non-essential, others must come from food called essential amino acid. Essential amino acids are 9 in number. For example, leucine, valine and lysine. Non-essential amino acids are 11 in number, for example, alanine and glutamine. Second one, the acidic and basic amino acids. Acidic amino acids have negatively charged side chains, and that is due to an extra carboxyl group. The acidic amino acids contain less amino groups and more carboxyl groups, for example glutamic acid and aspartic acid. The basic amino acids have positively charged side chains due to an extra amino group. The basic amino acids have more amino groups and less carboxylic group. For example, in lysine and arginine. Third and the last one, polar and nonpolar amino acids. Polar amino acids have hydrophilic R groups that form hydrogen bonds with water, making them water-soluble and typically found on protein surfaces. For example, serine, threonine, and asparagine. In contrast, nonpolar amino acids possess hydrophobic side chains that avoid water, instead clustering together in protein interiors to minimize contact with aqueous environments. Notable examples are leucine, valine, and phenylalanine.
Protein exist in many forms and shapes. Some have primary structure, some also exist as secondary, tertiary, and quaternary structures. All these forms are maintained by molecular interactions. In this video, we will describe the types of interaction that hold protein molecules in shape. The interactions are A. Hydrophobic interactions, B. Hydrogen bonding, C. Ionic bonding, D. Covalent bonding, including disulfide bonds. E. Van der Waals forces. Let's get the details. First, the hydrophobic interactions. These are weak, non-covalent interactions between nonpolar amino acid side chains that cluster together to minimize contact with water. Second one is hydrogen bonding. Hydrogen bonding. This occurs between the electronegative atoms, oxygen or nitrogen, of one amino acid and the hydrogen atom of another amino acid, forming a weak yet important bond that contributes to protein structure. Coming up next is ionic bonding. Ionic bonding. This occurs between oppositely charged amino acid side chains, e.g., positively charged lysine with negatively charged glutamic acid, creating an electrostatic attraction. Covalent bonding. Here we are taking the example of disulfide bonds. This is a strong bond formed by the sharing of electrons between atoms, and disulfide bonds specifically form between cysteine amino acids, playing a crucial role in maintaining protein structure. Van der Waals forces. These are very weak, temporary interactions between molecules due to fluctuations in electron distribution. While individually weak, they can contribute to the overall stability of protein structure when numerous occur. These all interactions maintain the shape and nature of proteins. We have already discussed what are protein made up of. Now in this video we are going to discuss the levels of organization of proteins. Here we will discuss the primary structure of protein, secondary protein, tertiary protein, and the quaternary structure of protein with examples. First one is the primary proteins. As you can see the picture of primary proteins, they refer to the basic, linear sequence of amino acids that make up a protein. This sequence is determined by the messenger RNA. It is very crucial because it dictates the protein's three-dimensional shape and ultimately its function. Changes in the primary structure. Even a single amino acid alteration can significantly impact the protein's ability to perform its designated task. Some examples of primary protein are insulin, which is a hormone whose primary structure is a linear 2 polypeptide chains. First 6 amino acid chain in hemoglobin is also linear primary protein. Next is the secondary structure of protein. Secondary protein structure refers to localized folding patterns within a polypeptide chain, stabilized primarily by hydrogen bonds between backbone atoms. The two most common types are the alpha helix, which is a coiled structure, and beta sheet, which is a zigzag, pleated strand. These structures provide stability and contribute to the protein's overall 3D shape. An alpha helix is a coiled, right-handed spiral structure where the polypeptide backbone forms hydrogen bonds between every fourth amino acid. While a beta sheet is a sheet-like structure formed when multiple polypeptide segments lie side by side, connected by hydrogen bonds. These are both common types of secondary structure in proteins, contributing to their overall three-dimensional. Examples of proteins with secondary structures include keratin, found in hair and nails, and collagen, a fibrous protein in connective tissues. Tertiary protein structure refers to the three-dimensional folding of a single polypeptide chain, driven by interactions between amino acid side chains, R groups. These interactions include hydrogen bonds, ionic bonds, hydrophobic interactions, van der Waals forces, and disulfide bridges, which we have already discussed. Examples of proteins with complex tertiary structures include enzymes, antibodies, myoglobin, and hemoglobin. Next, the quaternary structure refers to the arrangement of multiple polypeptide chains into a single functional protein complex. As you can see in the picture, the tertiary proteins has a single polypeptide chain, while quaternary protein are made up of 2 polypeptide chains. Examples are hemoglobin, a tetrameric protein in red blood cells that binds and transports oxygen cooperatively. Tetrameric means it, made up of 4 polypeptide chains. 2 alpha and 2 beta chains. DNA polymerase have multiple subunits working together for DNA replication. Antibodies are composed of 2 heavy and 2 light for antigen recognition. Let's differentiate fibrous protein and globular protein. Fibrous proteins are elongated, structural proteins, and often water-insoluble. They are primarily responsible for providing strength, support, and protection to tissues and cells. Some major examples are collagen. It is a major component of connective tissues like tendons, skin, and bones, providing strength and flexibility. Keratin, which is found in hair, nails, and skin, contributing to their hardness and flexibility. Elastin, that provides elasticity to tissues like arteries and ligaments, allowing them to stretch and return to their original shape. Fibrin is a key protein in blood clotting, forming a meshwork to stop bleeding. Myosin, a protein found in muscles, responsible for muscle contraction. While globular proteins are spherical, water-soluble proteins with a compact, folded structure crucial for their diverse biological functions. They are characterized by a relatively unstable structure due to weak non-covalent interactions, making them sensitive to environmental changes like pH and temperature. Common examples include enzymes like amylase and lactase, hormones like insulin and testosterone, and transport proteins like hemoglobin and myoglobin. Antibodies, also known as immunoglobulins, are another important class of globular proteins. That's all, students. Don't forget to subscribe the channel.